Hi everyone, my name is Andy Pang. Today I'll be talking about using optical genome mapping for genome integrity QC. So here is an outline of the talk. First, I'll introduce the various OGM workflows for various variant allele fraction VAF sensitivity. Then I'll be sharing with you some additional results from the NIST Genome Editing Consortium. And I'll follow up with some more sensitivity performance using a dilution series. And finally, I'll be talking about how to actually use OGM for the cell and gene therapy application and the actual workflow. So when we are talking about bioprocessing QC, we are looking for on-target events such as the edited gene validating the edits, as well as off-target events, that is to screen for genome instability as a result of the editing. Currently, users can use chromosomal microarrays to look for genome-wide copy number variation. But as we know, microarrays cannot detect balanced events. Users can also use karyotyping to look for large rearrangements, but with karyotyping, we are limited to only observing very large rearrangements. The technique is labor-intensive and difficult to scale, and it requires expert cytogenetic technician to do the analysis. With optical genome mapping, OGM, it can potentially meet both analysis need on a single platform. It is high resolution, can detect events down to 500 base pair and up. It can verify the position of the transgene insertion. It can detect all classes of structural variants, balanced or unbalanced. It is efficient, scalable, and it has an automated variant calling and comparison tool. And it also has a fast turnaround time. So for the cell QC applications, we can be looking at doing QC for bioprocessing, cell therapy, immune cell therapies, or for research, such as looking at the integrity of immortalized cell lines. So the table here, I'm showing you the workflows according to what you're looking for. Suppose you're looking for verifying the major clone that it is correct, and you can use our germline workflow, which is aimed to detect SVs whose variant allele fractions 50, 100%. One would need to collect 400 gigabase pair of data or 80x effective coverage. If you're looking for SVs down to a variant allele fraction down to about 5%, you will be using our somatic workflow. It has sensitivity down to about 5%, and you would need to collect a total of 1.5 terabase pair of coverage or 300x effective coverage. If you're looking for something even more sensitive, low allelic fraction, based on preliminary data, when you can collect even more data, up to a total coverage of 5 terabase pair or 1200x effective coverage, that would enable you to identify calls down to 1 to 2% VAF. On the table here on the right, I'm showing you the limit of detection on the various SV size and type that can be detected. By the de novo assembly pipeline applied on the germline workflow, one can identify insertions and deletions 500 or 700 base pair and up, 50 kb for inversions and duplications and translocations. With the rare variant pipeline software applied on the somatic workflow, one can identify at 5% VAF insertions and deletions 7 kb and up, as well as about 70 kb and up for the larger rearrangements, for the other type of rearrangements. And again, based on research preliminary data, the ultra-rare variant workflow enables you to identify at 1 to 2% FAF, insertions, deletions, 20 or 50 kb and up, and 100 kb and up for the other types of SVs. Now, using the copy number algorithm, essentially examining changes in the depth of coverage to infer aneuploidy, the limit of detection there is between 5 to 15% VAF, depending on the chromosome size. Now I'm going to switch gear and revisit the NIST Genome Editing Consortium that Samantha Maraj just presented to you. And just to reiterate, the purpose of the consortium here is to test the assay's capabilities to report variant size and frequency. So they did a mixture of well-characterized genome in a bottle samples. The variants of interest range from single nucleotide variants to insertions and deletions at least around tens of kb. And the variant frequency ranges from about 0.1% to 50%, actually as shown here on the right. So the participants were given a list of genomic locations, but we were not told which genomes were used, what were the variants, as well as what were the expected frequencies. So we were given 5 samples that's not labeled of the expected frequency. We were asked to process the samples using a normal operation, and this here is to simulate detecting variants after genome editing. On the bottom right here, I'm showing you the participants of the technologies of the participants. And you can see that optical genome mapping is the only genome-wide technology which can be very useful in detecting any off-target events in cell QC applications. So what we did, we chose the deep coverage workflow. We collected 5 terabase pair of data, and we called SVs using the default rare variant pipeline. And the results are shown here. On the left, you're looking at the performance of detecting variants at least 5 kb and up. And the x-axis here shows the 5 levels of variant allele fractions. It turns out that OGM can detect 12 out of 14 of the expected large insertions, deletions from 5% FAF and up. The 2 that were missed are complex events, and they're under further investigation. You can also see that we have sensitivity in detecting variants between 0.5 to 2% FAF, and we also have sensitivity even at the 0.1 to 0.25%. Now, at the same time, on top of using the default rare variant pipeline to call SVs, we also used a molecule mapping approach, which we call genotyping approach. Essentially, what we did was we aligned the molecules from these latter 3 samples to the maps in this sample. And the idea is, since the maps here capture structural variants of interest, when we align the molecules to these maps and we observe coverage, then that is essentially telling us that the SVs is actually present in the sample, just wasn't called by our default pipeline. And using this genotyping approach, we're able to recover additional 9 variants, and we have even good sensitivity down to 0.1 to 0.25%. So what we are looking at here is the sensitivity of detecting polymorphic structural variants. We did additional analysis on larger variants, such as large deletions, duplications, translocation, or large insertion events. We also simulated trisomy events. And the results shown here with VAF from 50% to 0.5% and our performance. You can see that we have the vast majority of the structural variants. The limit of detection is somewhere between 0.5% to 2%. Now, but for trisomy or aneuploidies, the copy number depth of coverage algorithm, the limit of detection is somewhere between 5 to 10%. Next, beyond sensitivity, I'll be talking about how do you actually use OGM for cell and gene therapy, the actual workflow itself. I'll start with experiment design. So suppose you have a test sample, an edited sample, and a paired control, let's say the prenatal cells. You would collect data. Again, the amount that you would collect depends on your expected variant allele fraction, the sensitivity you want. After you collected the data, you will call structural variants for both the samples, run the dual variant annotation pipeline to compare the two call sets in order to identify variants that are unique in the test sample and not in the control, as those could represent the off-target rearrangements as well as the on-target insert or edits. As for sample preparation and data collection for the deep coverage workflow, you would still use the standard reaction of the sp-DNA isolation protocol. You would need twice the reagent amount required for the labeling and staining steps. And then you would add the solutions to three flow cells. And then you would put that into the Sapphire instrument to collect data to a target total coverage of 5 terabase pair. I'm also showing you here the time that is required for each step. So after you collected the data, you would, for both the test and the control samples, you would call SVs and run the dual annotation pipeline to compare the calls. And then our Bionano Access visualization software enables users to quickly identify, to filter for variants that is unique in the treated sample. Specifically, you can filter for SVs that are absent in the control SV call sets, as well as the SV molecules, as well as the control molecules. And in doing so, applying these filters, you can then quickly narrow down to a handful of candidates' structural rearrangements that could be treatment specific. And then you would then need to do manual inspection of the calls. Optical genome mapping is a very robust workflow. It has very low false positives. However, since we are potentially looking for something that's very rare, it is crucial to do these menu inspections. In the next few slides here, I'll be talking about what to look for during these inspection steps. First, we should look for false alignments. So in this picture here, you can see that this map calls a translocation between the top chromosome and the bottom chromosome. But if you look closely in the region, you can actually see there's an additional map, a longer map, a bigger map with more labels, but is aligned fully to the top chromosomes, just with an inverted alignment. Examining the label patterns, you can see that this fragment has the same label pattern and is essentially just a subfragment of the top map. So we would say that this translocation is an artifact due to misalignment to the bottom chromosome. The whole thing should have just been represented by the top map with additional labels. So no translocation in this case. Another artifact to look out for is very low variant allele, very low allelic fraction in the control cell lines. So this map here is deemed by the software as treatment specific. In the treated sample, there's a large insertion captured by this map. It has strong molecule support by the treated sample molecules. But if you were to look closely in the molecule pileup in the control sample, you can actually see that there are 2 molecules supporting the call. So by manual inspection, we can see that this insertion is actually present, but only in a very low fraction in the untreated control sample. Finally, I want to show you a true treatment-specific event. This map here calls a translocation between chromosome 1 and 11. It has nice, strong alignment, good molecule support, and importantly, there are no control molecules that support the translocation structure, no molecules that can span across the junction. So in summary, optical genome mapping enables quality assessment for cell processing. It is a robust method for detecting structural variation, can identify variants from 100% down to 1 to 2%. But the depth of coverage CMV calling algorithm may not be able to call very low VAF events. The technology can easily identify on and off target variants in edited samples. It has an automated software to do the comparison with the control sample. But we emphasized that it is still important to do manual inspection of candidate variants. And much of what was discussed in this talk here can be found in our white paper, which you can get on our websites. And with that, I'd like to thank you for your attention.